what sort of COVID vaccination is learning from influenza vaccination, and sort of vice versa, and where we are with the status of each, how COVID-19 pandemic has affected circulation of flu viruses, and then come back with a little bit of perspectives at the end. And then I think we're going to leave hopefully quite a bit of time for some questions. So if we look at these two viruses, we have on the side in red the SARS-CoV-2 coronavirus, so the causative agent of the COVID-19 pandemic, and here a representation of flu. If we're thinking about vaccination against these viruses, our approaches are actually pretty similar. So our vaccines against the coronavirus target this surface glycoprotein on the virus, the spike protein. On the influenza virus, most of our vaccines target this protein here, the hemagglutinin. And these are the two proteins on the surface of these two viruses that attach them to the host cell, get them inside the host cell, and start the replication of these two viruses going. And so from a very high level overview, our approach to vaccination against these two viruses is pretty similar. What is very different at the moment is how these two different vaccines actually present those glycoproteins to the host immune system. With influenza, we're essentially delivering that vaccine antigen, or that hemagglutinin protein, to the immune system in sort of native virus form. So just like that in most cases, just like that protein sits on the surface of a virus. And the technology we use for influenza vaccines is actually quite old technology in most cases. Some of the earliest forms of flu vaccines were done in the mid-1940s, and they in many instances haven't changed a whole lot since. But there are a number of different formulations. If we look at the one that's perhaps a little bit different, is this recombinant protein version of the flu vaccine. This is a vaccine that's licensed in just a few countries, where that hemagglutinin molecule is expressed in baculo systems, and then that is sort of the basis for vaccine. But a lot more traditionally, these other cases, so the live attenuated vaccine, we essentially grow up the influenza virus, it expresses that hemagglutinin on the surface, and we present that to the immune system in the form of a live attenuated virus or some inactivated virus. And there are different forms of that, either the whole inactivated virus itself, we split it up with detergents, or we even separate it into some of these subunits as well. So each of these is a fairly, with the exception of recombinant HA, is a fairly crude way of immunizing people. But it's an approach that's worked, and it's an approach that's served public health very well for a number of years. If we look at the approaches that we've taken, we sort of the global public health community, for immunizing people against the COVID pandemic, we've used much more modern technologies to again present, in this case, that spike protein to the immune system. I think a lot of sort of the early products on the global market were these messenger RNA vaccines, where essentially we're delivering that spike, not the spike protein, but the gene encoding it into the host cell, inducing antibody formation. There are some approaches that have used, again, different ways of expressing that spike protein in vitro systems, and also a lot of vaccine approaches that are using viral vectors to get the gene expression, that spike protein, into host cells, again, where it produces a protein and induces immunity. So again, if we're comparing and contrasting flu and COVID vaccines, the COVID vaccines are using a much wider diversity of platforms, and those platforms are, in many cases, far more sophisticated than what we're currently using for influenza. And we'll come back to this a little bit at the end, because I think one of the consequences of a global response to the COVID pandemic and vaccines is, I think this will change the way we also think about immunizing against influenza in the coming years. So again, coming back to the similarities of these vaccines, both these vaccines target these surface glycoproteins. They want to induce these antibodies here that block that interaction between in the case of the spike protein in the coronavirus and the ACE receptor on the human cell. In the case of influenza, block that interaction between that hemagglutinin protein and sialic acids in the host cell. But again, this primary mechanism is essentially the same between these two vaccines, although in principle they're certainly different, quite different ways of going about it. The main problem with that is by targeting these two proteins, either the spike or the hemagglutinin protein, these are the two most variable proteins on both of those viruses. You know, and from the context of immunization, that's not a good thing, right? So we're targeting the most variable part of those viruses, and we know from a century of understanding the evolution of flu that these proteins change over time. So we get antibodies that block this interaction, the virus mutates, and we lose a lot of this immunity. Therefore, we have to update vaccines, at least for influenza and possibly for SARS-CoV-2 as well. And about 70 years ago, the World Health Organization realized that to have a successful vaccination campaign and successful flu vaccines, they had to put in place a global monitoring system. And the system that has developed over the past, I think maybe it's next year will be the 70th year of this program, which is now called the Global Influenza Surveillance and Response System, or the GISRIS. So this is a network that has now over 140 national influenza centers around the globe, including in Thailand. There are 6 collaborating centers in this network. So the lab that I am at St. Jude in Memphis, Tennessee is one of these collaborating centers, and then 4 central regulatory laboratories. And the primary purpose of this network is to monitor influenza viruses during the respective flu seasons in these countries, characterize those viruses, and then bring all that information together twice a year to make recommendations for influenza vaccine composition. We meet once in February to make recommendations for the Northern Hemisphere formulation, and once in September for recommendations for the Southern Hemisphere. And if we look, we basically come together with all this information collected from these laboratories and ask a pretty simple question, which is, are the circulating viruses similar to what we have in the vaccine already? And if the answer to that is yes, then we don't change the vaccine. If the answer to that is no, then there will be recommendations to change the composition of our vaccines. And there are multiple data streams that come into making this decision, answering this question. We look at both human and zoonotic flu viruses. Over time, the system has changed a lot, and certainly now there's a lot more sequence information available with next generation sequencing platforms. Of course, it's much easier to generate the sequence information now. So there are thousands of viruses that are sequenced a year. The most important information is the antigenic information, however. This is done with much lower throughput assays, either hemagglutination inhibition or microneutralization assays. Again, these ask the question as, from the point of view of antibodies, have these viruses changed and how antibodies recognize them? There are some human serology data done. There's some predictive modeling that is now being brought into this decision as well. These models are still learning, they're still being developed, but I think will become more important over time. And we also look at vaccine effectiveness data as well. So look at the previous season, how well did these vaccines do? It's actually quite interesting, and this is not necessarily anything to do with vaccine, but we actually met in the end of February of this year to make these recommendations for the upcoming Northern Hemisphere influenza season. I want to share a few slides of this with you because it's quite incredible what the pandemic has done to influenza activity globally. So here, if we look at so this is the week of the year starting in January here going through to December. Over the last few years, so 2018 in green, 2019 in blue here, you can see pretty even numbers of samples that are characterized throughout the year, peaking at just under 200,000 samples a week that are characterized through this system. You can see there was a little bit of a peak early in 2020 with the beginning of the COVID pandemic, so more samples were coming into the system, and that continued through the rest of the year. Dropped off a little bit towards the beginning of 2021. But I think the most surprising and unexpected outcome was when we actually looked at how many of these samples were positive for flu. Again, if we have a look at historically, 2017, 2018, 2019, we see these peaks of positives again during the Northern Hemisphere winter season. But if we follow the red line here, which is 2020, so we started off, flu season was just kicking off, then the pandemic came along, and then so the percentage of influenza positive samples plummeted and essentially stayed low all the way through the Southern Hemisphere winter season, and again all the way through the Northern Hemisphere winter season as well. So I think it's pretty well accepted now, but this is actually some numbers behind it. When the essentially coronavirus pandemic came along, influenza disappeared. We don't exactly know why this is. Clearly mask wearing, social distancing had an impact on that, but I'm not convinced that was the only thing as well. I think there's probably a little bit of viral interference went on as well. And it wasn't just flu that went away, so a lot of other respiratory viruses disappeared as well. This was a study I was involved in actually in New Zealand, which is my home country, looking at a number of other respiratory viruses. So RSV, of course, is another big winter disease. The green line here is sort of the average activity over weeks. If you had the blue here is essentially the period of lockdown in New Zealand when they locked down at the beginning of the COVID pandemic. So typically you would then see RSV activity pick up, but there was no RSV activity during this period in New Zealand. There was very, very little enterovirus, very, very little adenovirus. About the only virus that was causing respiratory disease in New Zealand at the time was actually rhinovirus. So a lot of other metapneumovirus, the parainfluenza viruses all went away as well. So I think a very, very unexpected consequence of this pandemic has been what impact it has had on the circulation of other viruses as well. And of course, this has caused a lot of challenges for picking the right flu vaccine for the upcoming Northern Hemisphere season. There have been very, very few viruses with which to help make that prediction. So I guess we'll have to see coming up what strains of influenza and these other respiratory viruses will emerge. If we look at how effective flu vaccines are, we know that they're variably effective, and this is I apologize, this is not the best of slides, but when we look at vaccine effectiveness historically, and these are all just different estimates of the efficacy of H1N1, the component of the H1N1 of the flu vaccines, and this is 50% efficacy, this dotted line down the middle. So, typically the point estimates for efficacy of flu vaccines hovers round about the 50% mark. For the H3N2, it tends to be a little bit worse. So flu viruses work, they're a valuable public health tool, but they aren't the best vaccines we have. So in comparison, sort of now switching to the COVID vaccines a little bit more, you know, most of these COVID vaccines in comparison are really superb vaccines. And, you know, we're now starting to get a lot more of this effectiveness data out from the clinical trials. This is just some of the earlier ones. There's clearly more information out there than this, you know, and they range from sort of the 70% from the adenovirus-based vaccines, which is the J&J or the AstraZeneca, you know, right up to the high, mid to high 90% effectiveness for some of the mRNA vaccines, the Moderna or the Pfizer, or some of the protein expressed like the Novavax as well. So again, these modern technologies have done, at least in my mind, incredibly well, and certainly better than I would have anticipated. Others aren't quite as effective. So this Sonovac is a Chinese-produced vaccine. The technology made to use this is very similar to the technology we use to make flu viruses. Essentially, it's the virus grown up and inactivated. So again, these older, more conventional technologies don't seem to be as good as some of the newer, I think, more sophisticated approaches. And we know, so this is actually just some data from our hospital. So at St. Jude, the hospital I work at, since about March of last year, we've been swabbed at least once every 2 weeks as we come into work. And starting from about November of last year, we started to get vaccines started coming into our population, and we started to sort of track how vaccination is going in our population. If we look at sort of cumulative incidence, so the percentage of the tests that have been positive sort of over time, cumulatively, if we look at people either symptomatic or asymptomatic, we see it slowly climbed with the unvaccinated group. But if we look at those that have been vaccinated, again, it's very leveled off right now. And again, we're detecting very, very few infections in our vaccinated population at our hospital. And that goes with both symptomatic and also the asymptomatic as well. So we do detect a number of infections in asymptomatic people just walking onto campus. But there's quite a difference again, even in that the vaccine is doing a really, really good job of protecting from asymptomatic infection as well. So again, really encouraging data, I think, from a number of different sources. Of course, there's rapid progress. Of course, there are some big questions still to come with our COVID vaccines. You know, there are manufacturing and supply issues, and which I think is unfortunately manifesting in sort of a global disparity. You know, I'm almost embarrassed to say that certainly I think where I am in the US, you know, very soon we're going to have more vaccine than people willing to take it. So in our local community, we're at about a 30%. 30% of the population have been fully vaccinated. And again, I think we're starting to get to a point where a lot of people left may not want to take vaccine. So we really need to think hard about how we get some of the spare vaccine to other parts of the world that really need it. The duration of immunity is another big thing. You know, most of these vaccines require 2 doses. You know, are we going to require a 3rd or a 4th to boost that immunity up? And of course, the other big thing is that these variants are emerging, right? So we've all heard about these variants. So much like flu, where flu changes, we have to update the vaccine. We're now starting to see the SARS-CoV-2 virus change. At this point, we're not sure if we're going to have to update the vaccine to chase it, but it's looking a little bit like that potentially could be the case. Just another little bit of data that we've been generating in our lab with, this is data that's coming out of hamsters. This experiment hasn't actually finished yet, so I apologize, it's a little bit rough, the figure, but I just wanted to share it. So in this experiment, we actually vaccinated hamsters with an mRNA vaccine from some of those early Wuhan-like variants, or not variants, but Wuhan-like strains of the virus. And then we challenged those animals with, in this case, the B117 virus, which is the UK variant. This is probably one of the most dominant strains around the globe now. In the US, it's our most dominant strain. If you look here, these are the vaccinated animals in the pinkish color, and the purplish color of the unvaccinated animals. So you can see this vaccine, the old vaccine, is doing a really, really good job protecting against the UK variant, and a similar type of thing against the South African variant, this B1351. This one is a little bit, has a few more changes in antigenic sites in the UK variant, but we're still seeing some, again, we're still pretty early, we're still seeing some good protection against this virus as well. So at least, and I think certainly there's other data coming out like this as well, at least in animal models, it looks like our current vaccines are doing a reasonable job against some of these variants as well, even though in sort of laboratory, more serologic tests, it looks like they are changing. Just checking my time here. Okay, so I'm just gonna close up here with a few conclusions. So basically, if we're talking about flu and COVID vaccines, the basic immunologic principles underlying how we vaccinate against these two viruses is pretty similar. But COVID vaccines really have the edge in terms of the sophistication of the approaches we're taking. And I really think, particularly this mRNA technology is going to change the way we make a lot of vaccines against infectious diseases, including flu. I think it's probably going to see an end to the way we vaccinate against flu now. It won't be till next year or the year after, but in time. We know we're going to continue to update our vaccines against flu. We don't know yet if we're going to have to do the same against COVID. There are some people who think we definitely will, others who think maybe not, and some people in between. So only time's going to tell there. And the other thing that we're definitely going to need for SARS-CoV-2 in the meantime is one of these GISRIS-like surveillance systems to monitor that virus on a global scale. And these systems are being developed now, both at the WHO level and at national levels as well. So lots more to come on that. Lots of systems that need to come into place before we can answer some of these questions. So I'm going to finish there. Lots of people involved in this, and I thank you all for listening and happy to take some questions.
Thank you, Richard. It's very nice presentation. So we have six minutes left for the question. If anyone has any question, please raise your hand. Just raise your hand in the Zoom meeting. Okay, maybe there are some questions from our audience. There is an article from EMA discussing the possibility of blood clotting condition after receiving adenovirus COVID vaccine. Would this possibility be able to happen in the case of people being vaccinated by mRNA vaccine?
Yeah, so I mean, it's interesting data, right? So it's in terms of at least the data from the United States, it was about, well, certainly less than 1 in a million, and it was all in middle age, well, not middle age, but younger female. So it was a a definite portion of the population that had it. There have been a couple of clotting type episodes seen with the mRNA vaccines, but not at the same frequency. So it does seem to be associated with those adenovirus platforms, but not exclusively. And again, the risk of that clotting is very, very low, and certainly the risk of coming down with severe COVID is far higher. So the risk benefit to the vaccination is clearly in favor of getting vaccinated.
Okay, thank you. There's a question from Dr. Tirasat. You can speak now.
Hello. Thank you so much, Dr. Webby. It's a great presentation. I have some questions. As we know that the pharmaceutical company announced that the efficacy of the vaccines are very effective. But as you know, they have a different aspect to evaluate the efficacy of the vaccines. I just wonder, do you have any information to make sure that everybody who have the vaccination will have the neutralizing antibody raised up or something like that? And if somebody don't have the neutralizing antibody, you think what is the reasons?
Yeah, I mean, we have a couple of studies that were involved in, sort of look at that. And at least with the mRNA vaccines, everyone that we've looked at has responded to that vaccine. Of course, they have different levels of neutralizing antibodies. I must admit, I'm not sure of the clinical trial data, the big trial data, whether they identified individuals who didn't seroconvert at all. Certainly with influenza, we know that happens. People don't seroconvert. I think it's far less with these COVID vaccines, just because they're inducing a much more robust response. But we know with all vaccines, right, that different people are going to respond differently to vaccines. Some are going to produce more neutralizing antibodies than others. And it's possible, I think, if we can identify those individuals who haven't done as well, there are more, well, theoretically more possibilities, right? So perhaps if people didn't respond as well to the adeno, they should be offered some mRNA vaccines. I understand in many places of the world, there aren't choices, right? So you take what you can get. But no, I think it's an interesting question. But I think it seems to be pretty good.
Okay, I just like to ask for more opinions from you. Since we know that some people will not produce some neutralizing antibody, and many people talk about the mutation of the virus that will be the problem to make a neutralizing antibody. Do you think it's possible that there is other factor like a host factor, like a genetic of different people, about the HLA or something like that?
No, I think absolutely. I think there is some emerging evidence and pretty strong evidence that host genetics absolutely influences how some people respond to vaccines. And so, yes, and I think there's work going on in that area as well. So no, I absolutely agree. Host factors definitely play a role in response to vaccines for sure.
Yeah, thank you. I think we have more further study about that. Thank you so much.
I agree. Thank you.
Okay, there are last question from our audience. How far the available COVID vaccine now have gone through complete clinical study?
That's a good question. So how many have gone through? So again, from a very US perspective, because that's kind of where I am, I know there have been 3 licensed in the US. So the 2 mRNAs from Moderna, Pfizer, and the adenovirus from J&J. Certainly the AstraZeneca product has been licensed in a number of countries as well. There is also a number of products out of China. The Sonovac that I talked about has been widely used as well, widely distributed, and certainly some of at least 1 out of Russia as well. So we're talking at least licensed in at least 1 country. There are at least 6 vaccines that have gone through at least that phase 3 trial and have been, not all of them are fully approved, but at least approved for at least emergency use. So, I mean, it's an incredible, the fact that we're sitting here, just over a year into this pandemic, and we have, 6 vaccines that are in use in people around the globe is just an incredible public health response. And it's amazing in my mind.
Okay, thank you. Maybe we will finish for this section,
Thank you.
but